Hi everyone, it's Mr. Sinti, and today I'd love to talk to you about enzyme regulation. That's right. Enzymes are protein, often catalysts that speed up chemical reaction or speed up or regulate metabolism in cells. And so this particular video is going to look at some of the important factors that influence the activity of enzymes. And those could be physical phenomenon. In other words, like concentration of substrate itself, or the concentration of the enzymes, or it could just be the environment that the enzyme's working in. So we want to consider things like the amount of salt in an area, the pH, the temperature, and even some local environments are conducive. And there's even some molecules that influence the activity of how well an enzyme is working. And so let's jump right into this conversation because I think it's going to be very good. Um, it's one thing understanding how an enzyme works, but how what the factors that regulate it is perhaps even more valuable, as we shall see. So check this out. Say you had, like, I know this seems like a silly thing, but roll with me on this. Say you had a bunch of toothpicks right here, and this was you as a student, and your activity was the chemical reaction in question was that you were to break toothpicks, and so what you would be asked to do is, in other words, like take them in your fingers and snap them into halves like this. So you're breaking them up. That's the chemical reaction. And so if you were asked to do this, perhaps your hands would be acting as an analogy of the enzyme. And so if the substrate's a toothpick, maybe you're a toothpick ace. And so here you are breaking the toothpicks up like this, okay? And so what's up with the timer on the cell phone here? Well, one could look at the rate at which toothpick ace is capable of doing this. And how would you go about doing that? Well, you could look at the disappearance, and this goes for all chemical reactions. You could look at the disappearance of substrate over time. That's a rate. Okay, we're talking about rates now. The rate of the reaction, the rate of the reaction. Or you could look at, conversely, it would be the same thing, the appearance of product. In other words, you'd have to count that over a certain amount of time. So the appearance of product over time. And so if you monitored this, in other words, like you took tally of it every 10 seconds, every 15 seconds, you would be able to throw up a graph. And so let's consider this. So, like for example, if you had over here, this is the substrate. Do you remember toothpicks like that? And so this would represent, let's do this, substrate. Let me write that in. Substrate concentration. Substrate concentration. And do you see here, this would be increasing in this direction on the x-axis, like that. So substrate concentration. And this over here would be rate of the reaction. Now, what would this look like? What would this look like? Well, picture yourself again back in that little tub, and you're blindfolded so that there's no bias. Your hands are just sort of searching around for substrate. So when you find one, what do? You crack it. And so how would you like this? So picture yourself breaking the toothpicks. But if I were to measure this, okay, and it might be looking something like this. Look, the rate of the reaction is going up. Okay, it's increasing, but check this out. If I were to add like a bunch of toothpicks, because think about it, as you're breaking the toothpicks, you might be searching, like where's the, oh, there's one, or oh, there's one. If I dumped a whole bunch of toothpicks into that little tray, that might make it easier for you as an enzyme to be able to find the substrate. So check this out. Perhaps, watch this, as you increase substrate concentration, the rate of the reaction is increasing and increasing. Do you see this? How it's going faster because the enzyme is not even hesitating to be able to find a substrate. But if I keep increasing it and increasing it and increasing it, watch this. You think it would still go up and up and up and up and up? Interestingly enough, this is what happens. Okay, so the reaction's still proceeding, but the rate isn't increasing. So there's a point at which I can increase and increase the substrate concentration in a chemical reaction. And sure, that does increase the rate of the reaction, but then it reaches a point where it doesn't anymore. Can you think about why that might happen? That's an interesting thought. Okay, so where the rate of specific number of enzymes that convert the substrate to product depends on the substrate concentration. So the rate of the specific number of enzymes. So this is important because as you increase substrate concentration, as we were just saying, it makes it easier for the enzyme to be able to speed up the chemical reaction. So an increase in substrate speeds the binding availability to the active sites. And so there's no question that the enzyme's working at its maximum rate, as we saw just a moment ago. Let me just refresh you on that. So this is the enzyme maximum rate at this point, right here. But then it seems to, the reaction continues even though you're increasing substrate. Like, what's going on with that? Well, if you think about it, how many toothpicks can you break? I mean, you only have two hands, and so if you cut down on the possibility of finding them so they're readily available in the tray, you're like, okay, this is the max rate. This is the maximum rate of the enzyme. You could determine the maximum rate of an enzyme. But there's a limit to how fast a reaction can go. So that's very important. And so what we say is that there's some concentration of substrate where the active sites of all the enzymes in there are completely engaged. Like, I'm working as fast as I can. That's the maximum rate of the reaction. And so that's when the enzyme is said to be at saturation point. In other words, it has as much substrate as it can possibly handle. It's kind of a pun as far as the toothpick ace is concerned. So the only way to increase productivity is how then? If I want the rate to go even faster, and I'm like dumping in as much substrate as possible, you're like, well, many hands makes light work. And so maybe at this point, if I add more enzyme, it would speed up the reaction even further. So check this out. This is what we were seeing before with toothpick ace. So as you increase substrate, the enzyme's rate is increasing, but then you get to a point where it's going as fast as it can. So this is the maximum rate of the enzyme. You've determined it. If you want this reaction to go, let me switch the color. If you want this reaction to go even faster still, you're going to have to increase the number of enzymes in the chemical reaction. Okay, because if you just increase substrate, sure, it will increase, but then you reach a point where it's saturated. So this is the point of saturation. In other words, the rate isn't increasing anymore. So I hope that's clear. That right out of the gate, so that something that influences enzyme activity, of course, is substrate concentration and enzyme concentration. Those are two really important things. So now I want to emphasize, for the rest of this video, I want to talk about environmental conditions and what this sort of means. And so environmental conditions all has to do with the fact that the enzyme's ability to work has to do with its unique three-dimensional tertiary conformation. So the enzyme's forte is based on its structure. And so anything that would alter its structure is either going to enhance, let's keep it positive. It might even enhance the enzyme's ability to act as a catalyst, or it could be detrimental or deleterious. And so the environmental conditions will influence this. Here's a primary structure folding into the tertiary structure. And so any shape change, so this is crucial, I'm just going any shape change, any shape change in shape will influence the rate of the reaction. So if we have a shape change, and so what sorts of things can influence the shape of an enzyme is the question. And so there's certain conditions in and out of the cell that will optimize the rate of the reaction. And so this is the crucial thing. What's the best rate at which the enzyme will work? In other words, what's the best shape that we're trying to determine? And what's interesting about this is what determines the best shape? Well, there's a lot of variety, as it turns out. Not to go off track a little bit, but I think it's an important point to make, is that DNA ultimately is the genetic code for the polypeptide sequence, which ultimately determines the shape. And so you get different mutations that occur along the gene. And so you have different shaped enzymes. And so just like the morphology or the phenotype of an organism that is either more camouflaged or less camouflaged, the enzymes that are more optimal are going to be the more efficient one. And so natural selection accounts for enzymes reaching their optimal rate based on shape. I find that that's pretty cool. So one of the environmental influences, of course, that would affect the rate of a reaction is temperature. We know this because, like, let's look at this graph, for example, like the rate of photosynthesis, but it could be the rate of any chemical reaction. Do you notice here on the x-axis, as we increase temperature, the rate is increasing. Now, why is that? Well, the substrate is moving a little bit more quickly, the enzyme is moving a little bit more quickly, so it's accelerating the interaction between substrate and enzyme. And so that makes sense. Like the warmer things are, the more kinetic molecular energy is happening inside the reaction, and so the reaction rate will occur more quickly in an enzyme catalyzed reaction. So look at this. As you increase temperature, it's merrily going along, increasing the rate, and then, right here, it seems to be like peaking. Do you notice here, like this is the maximum rate at this particular temperature? I wonder what it is. And so as a scientist, we're always curious. This is the variable. What is the best temperature? What is the optimal temperature for this particular enzyme? Now, all the enzymes have different optimal conditions. That's something that you need to know. It's not every enzyme works best at body temperature. Some prefer cooler, some prefer warmer. And so it's up to us as scientists to determine optimal temperature. And how do we know it's optimal temperature? Because the rate is at its peak, but then look at this, it starts to decline. So it's not, even though you've increased the temperature, it's working only as good as it was before. And then watch that there's actually a decline. Seems odd to me. As you would continue to increase temperature, it was looking good here, and then it drops off. Any thoughts about why that might be happening? Well, temperature has a major impact on the rate. As temperature increases, as I was saying, that increases the collision rate between substrate and enzyme, and so the active sites are engaging the substrate at the maximum possible interaction. But then check this out. If you go too far, that increased, remember heat is like thermal agitation. If there's too much heat for a protein, it'll begin to stress the molecule out. And remember it's all about the shape. And so if you start to move the molecule, you might start breaking some of those weak bonds that are holding the tertiary structure together. You could be fracturing some of those hydrogen bonds or even the ionic bonds if it's moving very quickly. So if you affect the protein's active conformation, you might even denature the protein, and therefore it doesn't work efficiently at all. And as you can see just back here, too much heat and it's not working well. The molecule's shaking, and it's losing its shape. And if you went too extreme, you might actually cause the protein to denature completely, and therefore it won't function at all. So there's an optimal temperature. I really want to emphasize that. There's an optimal temperature that we're always looking for. So is 10 degrees Celsius optimal? And you would say, well, it's good, but it's not the best. 20 is better than 10. You're like, 30 seems better than 20. 37. Interesting. So this is optimal for most human enzymes, body temperature. What a coincidence. There is no coincidence. This is the atmosphere. This is the environment for which the enzymes work best. Again, natural selection has accounted for this. So even a slight fever of a few degrees could be dangerous, could even be lethal. If you take the temperature up to 40, 45 degrees, look out. You could agitate those enzymes so much that they're not working, and if all your enzymes start to break down, your chemical reactions will come to a halt, and therefore you might perish as a result of this. And so I want to point out that optimal temperature isn't always going to be around body temperature. There are some organisms, like for example, thermophilic, this sort of translates to heat-loving or heat-tolerant bacteria that we found in hot springs at Yellowstone National Park. Look at this. If you give them the body temperature, they don't do very well at all in terms of the rate of the reaction. They only do best at around, it looks to be like 80 degrees Celsius. And so it's fascinating to think, what are the conditions? In other words, what are the, like if you're talking about a mushroom that's sitting on top of a tree or a leaf, and it's decomposing it in the forest, what's the optimal temperature that those enzymes that are breaking down cellulose work best at? We're interested in these kinds of questions because if we want to bring this into the lab and optimize the temperature, because we might want to mass produce the enzyme and therefore break down cellulose for glucose and use it as a biofuel, we're very interested in what is the optimal temperature at which enzymes work. But certainly temperature affects the rate of the reaction. It seems to, as you increase temperature, it seems to work faster, again, because it accelerates the collision between substrate and active site. But then at a certain point, it doesn't work well. So the same thing can be said with pH. I want you to think of all of these conditions as doing the same thing. In other words, affecting the shape, influencing the shape. Because pH, again, whether or not your environment is just to refresh you, has a lot of proton or a lot of hydroxyl group. Okay, this will influence, these ions will influence the shape of the protein, because what's holding the shape of the protein together is the R group interactions, which are hydrogen bonds and ionic bonds and maybe even some covalent bonds. So there's an optimal temperature and there's an optimal pH that the enzyme works best in. And it's up to us as scientists to determine it. How do we know? Well, we alter the pH. And so, like, for example, as you can see here on the scale of pH, we're going from acid over here to base. Okay, so the numbers are going up. We can go like this, 7, 14. And so if we're looking at the rate of the reaction, we're like, the enzyme doesn't seem to work well in an acidic environment. Interesting. It seems to be working best in a neutral environment. For example, most enzymes sort of go along with this bell-shaped curve like this, and then apparently in an alkaline environment, it doesn't work very well. And so it's very important to maintain, if you're a cell, it's important to maintain that 7. You don't want to get too acidic or too basic, and so that's the need for buffers. These are, again, weak acid-base conjugates that help to maintain pH. So those are pretty crucial, because if your pH goes astray, perhaps your enzyme activity isn't very good, and therefore you're not very good. And so most of the enzymes fall in this neutral area. But then again, there's some very important enzymes that don't correspond to this. Like, I was just showing you like a typical one. A typical one might be like this, like around 7. Do you see that? In other words, it doesn't do well in acid or doesn't do well in alkaline. But check this out. These two enzymes are really important in our body. I don't know if you've heard of this enzyme. It's called pepsin. It's found in our stomach, our cells of our stomach, the mucosal layer, the epithelial. Those columnar cells secrete an enzyme, actually a precursor. They release an enzyme that is misshapen. It's called pepsinogen. And what's kind of interesting is that these cells called chief cells secrete this pepsinogen. And what happens is, look at this, in an acidic environment, what the acid does is change the shape. And so it changes it to a good working enzyme. Whoa. So that's pretty interesting. So the gene, ready for this? I won't go too far with this, but the gene that's coding for the primary sequence is deliberately making a dysfunctional protein. And so when the acid changes the shape, it changes it for the best. So this is the optimal pH for pepsin. And then what's fascinating is, you may know this, that when the food leaves the stomach, goes into the upper part of the small intestine. You're like, well, it's still acidic when it comes out of the stomach, but not until it reaches a big dose of pancreatic juice. And one of the things in pancreatic juice is sodium bicarbonate, which is quite alkaline. And it not only neutralizes the acid, but it actually takes it into an alkaline environment. And so some of the protein digesting enzymes, like for example trypsin, this is also a protein digesting enzyme, pepsin. These are both hydrolytic proteins. Trypsin works best over here in an alkaline environment, because that's where it is in the upper part of the small intestine. So that's kind of interesting. So each each enzyme has its own optimal pH. Now, here's something. Enzymes don't seem to work best alone. They seem to, like for example, a baseball glove. Remember, if you've watched this previous video, a baseball glove seems like it can catch a ball, but as it turns out, if there's something in the active site, or there's a non-protein molecule that's somehow embedded, or a ring will fit a little bit better if you stick something in there. Sometimes there's non-protein helpers that are found to affect how well an enzyme catalyzes a chemical reaction. And we call these non-protein helpers cofactors or coenzymes. Co means sort of with. So a cofactor, what's the difference between cofactor and coenzyme? Cofactor is something that's not organic. In other words, smaller things like minerals, like zinc. So an enzyme works best if there's a zinc molecule associated with it. So that's not a protein. Or iron, or magnesium. Okay, these are ions like this. This could be +3. It's 2. And then copper. So cofactors seem to be very important in whether or not an enzyme works well. A great example of this is one of the most important enzymes in the entire cell is an enzyme that replicates DNA. It's called, you might know it, DNA polymerase. And DNA polymerase really needs the cofactor of magnesium to work. It really needs Mg+2, or else it doesn't work efficiently. And so that's kind of curious. Now you're like, well, why is this? Well, it might be that these particular ions bind to the active site and therefore facilitate the substrate chemical reaction occurring more readily. It's kind of interesting how that evolved. Now, a coenzyme is same thing. It's a non-protein molecule, but it's a little bit larger than a cofactor, and it's organic, meaning that these are small carbon-containing compounds. Important example of coenzymes are vitamins. Now, vitamins, again, are not needed in the same quantity when you're eating as ordinary substrate food, like you're eating carbohydrate or you're eating protein, but you only need a little bit of vitamin. But vitamin is vital. You need it, or else you won't be able to survive. And so a lot of vitamins are coenzymes. They help enzymes. Now, enzymes are not needed in large quantities because they don't participate in the chemical reaction. So as it turns out, one of the coenzymes that is extremely important in biology in terms of the pathways and metabolism having to do with cell respiration is this guy called NAD+. It's a big name. It's nicotine amide adenine dinucleotide. And so we're going to see that these coenzymes are found in protein molecules in cell respiration, and they provide the ability for redox reactions to occur in that pathway. They're really, really important. Okay, so here's a couple of other molecules that either inhibit or help enzymes work. And so let's talk about inhibitors, for example. How would you like it if something, okay, this is normal right here. Here's an enzyme in purple, and here's a substrate that's supposed to fit very nicely to the active site. How would you like it if there was some sort of competitive inhibitor molecule? In other words, there's some molecule that's mimicking not just the substrate, but maybe just a portion of the substrate, so much so that it actually binds to the active site. This would be frightening because, again, this would prevent the substrate from binding to the active site. So this often is really, really dangerous. A competitive inhibitor could possibly even covalently bond, which means it's very difficult and often irreversible. And if it binds to the active site, it would be as if something was blocking your baseball glove and you weren't being able to catch baseballs anymore, or influencing your hands and you wouldn't be able to break toothpicks anymore. So competitive inhibition can be a real detriment. It can block the active site, and it can be lethal. And so, give you an example of this. You're like, well, what kind of things are we talking about? I mean, this cartoon drawing suggests that the competitive inhibitor could be large, almost as much as the substrate, but it could even be something really small. Like, for example, we might all know that lead is poisonous. We might know that mercury is poisonous. But why is it? Well, because these ions, even silver, these heavy metals have the ability to bind to the active sites of some crucial enzymes, especially in the thiol groups. They have an affinity for that. And so amino acids like methionine or cysteine can be influenced by that, and therefore the substrate isn't able to bind to the active site. Because as it turns out, a lot of the active sites have these thiol groups, or they have alcohol groups, or carboxylic acid groups. And so therefore, these heavy metals can be detrimental as competitive inhibitors. Now, competitive inhibitors are often dangerous, but sometimes enzymes have non-competitive inhibitors which are not dangerous, and they actually help by temporarily changing the shape of an enzyme. Now, this is rather crucial. If a molecule binds to not the active site, but another site, an other site is referred to as allosteric site. And if something binds to a protein over here, it'll ultimately alter the shape of the active site. Now, this could be very good because we talked about enzymes. Their ability isn't just to speed up chemical reactions. When the shape is perfect, then it optimizes the rate. But if the shape is slightly off, it doesn't work as well. And so maybe this is a good thing. Maybe you do not want a pathway to go as fast as possible. Think about that. And so one way to slow a pathway down is to temporarily alter the shape of the active site of an enzyme, and a non-competitive inhibitor is capable of doing that. And so it makes the active site unreceptive. And so this is critical. I put it in red here. It's reversible, and it is the way in which enzymes can actually truly regulate metabolism. Because the truth is, enzymes are incumbent to increase the rate of a reaction just by the nature of what they do. But if their shape isn't optimal, again, the reaction will go slower. And so therefore they regulate metabolic reactions. And so metabolic control often depends on this allosteric regulation. Again, if the active site is great, then the substrate will be produced into product. But if the active site is altered by an allosteric inhibitor, that will actually influence the shape of the protein, and so therefore it won't work as well. So these are non-competitor. They're not competing with the substrate. So they bind to another site that's not the active site. And often the allosteric inhibitor doesn't bond covalently, because then it would be permanently influencing the shape of the enzyme. So logically, this is probably a hydrogen bond over here, or possibly an ionic bond, probably hydrogen. So what's fascinating, are you ready for this? I've talked about it in terms of inhibiting. But sometimes, ready for this, sometimes a allosteric molecule can actually stimulate by sticking in. I make the statement that if you stick something onto a protein, it's going to detrimentally affect the active site. If you're following this discussion, you should say that you never know. You never know. So if something sticks, how about this? Let's go the other direction. If something sticks to the protein, maybe that encourages the active site to take a more optimal conformation. And so that's important to consider. So most of these so-called, quote, allosteric regulating enzymes are often at the beginning of a pathway, because that way you could sort of maximize and not be wasteful in terms of producing products that you don't want to. Most of these allosteric enzymes, in other words, enzymes that are capable of being regulated, usually are more than one polypeptide chain, so they're often they're often quaternary. Oops, let me go here. They're often quaternary. Okay, so this means that there's an allosteric site and an active site, and so they can oscillate between being really good and not being very good. And both of these are good, because sometimes you want the chemical reaction to go slowly, and sometimes you want it to go quickly. And so the protein oscillates depending on what the conditions are. So if you get something that sticks on here, that could actually open up the active sites. So these are called activators, as shown in red. if something like this, dark, which is the inhibitor, if this bonds to an allosteric site, it actually influences the active site so that the enzyme doesn't work as well. So again, both of these are good. Sometimes you don't want it to work well, sometimes you do want it to work well. So that's really cool. An example of something where you don't want to work well is something called feedback inhibition. So say you're making a product. Here's a pathway, a synthetic pathway, and there's a lot of this particular product. It can feed back within the cell and actually block up the allosteric site, changing the conformation of the protein, therefore less intermediate, less intermediate, less intermediate, and therefore the product goes down. And when the product goes down, there's less inhibitor, so then it produces more product. So an example of that would be this pathway to produce an amino acid, isoleucine. Now, if you're eating a bunch of isoleucine, that will actually block the first enzyme in this pathway. Likewise, if you're making a lot of it, it'll block the first enzyme in the pathway as well. So that's called feedback inhibition. Isoleucine does not compete with threonine. That would be the initial substrate. It binds allosterically and therefore inhibits it. That's pretty cool. So the product, when it's abundant, can actually influence this. And so here's something that's kind of neat too. If a substrate binds to an active site, sometimes the binding of the substrate alters the shape of the protein and actually makes it more receptive for other active sites to capture the substrate as well. An example of this is a great example of cooperativity. So hemoglobin is a great example of this. Once hemoglobin captures oxygen, the other heme groups are more likely to be able to adhere to oxygen as well. And then finally, the local environment within the cell helps enzymes as well. You may know that a eukaryotic cell, is composed of membrane-bound organelles, and membranes can actually inhibit whether or not ions are passing. And so different organelles have different degrees of acidity or alkalinity. And so there are certain enzymes that work best inside the mitochondria. There are certain enzymes that work best in the cytoplasm. And then finally, there's an organelle called the lysosome where there's hydrolytic enzymes, and those seem to work best in an acidic environment. And so that the lysosome works really hard to pump in protons because those hydrolytic enzymes have an optimum pH of acidic environment. So I hope this video on enzyme regulation and the factors influencing enzyme activity was informative and useful. Thanks for watching.